Good morning or good afternoon, everyone, and welcome to Genome Webinars. I'm Julia Caro, Managing Editor at GenomeWeb, and I'll be your moderator today. The title of today's webinar is Catch It If You Can, CRISPRa Tracing of Therapy-Resistant Cancer Cell Clones. This webinar is sponsored by Lexogen. Our speakers today are Christian Umkehrer, a scientist at the Research Institute of Molecular Pathology in Vienna, and Dr. Martina Sauert, product manager at Lexogen. You may type in a question at any time during the webinar, which you can do through the Q&A panel, which appears on the right side of the webinar presentation. We will ask our speakers your questions after the presentations have concluded. Also, if you look at the bottom tray of your window, you will find a series of widgets that can enhance your webinar experience. So with that, let me turn it over to Christian. Please go ahead.
Thank you very much, Julia, for the introduction, and welcome everyone. And in the next 40 minutes or so, I would like to tell you a little bit about CaTCH, which is a functional lineage tracing tool, which we recently developed in the lab of Anna Obenauf at the IMP in Vienna. And we use it to study the clonal evolution of resistance to targeted cancer therapy. So in the Obenauf lab, we are interested mainly in the successes and the unfortunate failures of targeted therapy responses. And one of our main models or research topics is the metastatic melanoma. And metastatic melanoma is frequently mutated in the BRAF oncogene, which is shown on the right here and indicated by this mutation to be mutated. And this mutation turns BRAF into a constitutive active form, which then leads to constant signaling of the MAP kinase pathway, telling these cancer cells to grow and divide uncontrollably. And therefore, specific inhibitors of BRAF have been developed, so-called RAF inhibitors, and these inhibitors block the signaling of the pathway in these alterated cells, therefore leading to the death of these cells. And in the clinics, these RAF inhibitors have been shown to be very effective, as we can see in this example of a patient harboring late-stage metastatic melanoma. And just 2 months of therapy, the tumor regressed in this patient, and he essentially was tumor-free. However, an unfortunate problem of targeted therapies, which is not only limited to RAF inhibitors and MAP kinase pathway inhibitors, but most targeted therapies have this problem, which is that they will eventually fail due to the emergence of resistance. And this is also what happened in this patient, who just 3 months later relapsed on therapy. And in this situation, the tumor does not respond anymore to the therapy at all, and there is no more treatment option using these targeted drugs. And to understand what is happening here on a cancer cell level or on a tumor population cell level, we can think of the tumor at the baseline stage of a large population of heterogeneous cells, so very different cells, where most of these cells will actually respond to the applied RAF inhibition. And this leads to this, and they will respond, so the responding cells will therefore die when the drug is applied, and this is what we see in this regression phase. However, a few cells are capable of surviving this selective pressure posed by the drug, and these will then regrow as fully resistant tumors not responding to the treatment anymore. And the resistance mechanisms, so the ways that these resistant cancer cells exploit to be able to grow in the presence of the drug, have already been extensively studied. And it has been shown that they commonly lead to reactivation of the targeted MAP kinase pathway. And this can be achieved via a wide set of different mechanisms, for example, through to expression of alternative splice forms of BRAF, but also amplification of BRAF or mutations in other pathway components, for example, in MEK or in RAS, but also alterations in secondary pathways have been described. And also more recently, it has been more and more prevalent that also epigenetic alterations are very common mechanisms that lead to resistance to these applied RAF inhibitors. So while the resistance mechanisms itself are already quite well studied, the question on how resistance originates in the first place is still largely debated. And there are essentially two main possibilities on how resistance might arise, where the first one is that resistance might actually be an acquired feature, while the second option might be that resistance could be pre-existing in certain cells. So in a pre-existing case, there might be a small population of cells residing within the tumor before the treatment is applied, and these cells already are fully resistant and harbor all of the necessary traits to be able to grow in the presence of the drug. And while the majority of the cells will then die when the drug is applied, these few fully resistant cells will survive and simply get selected by means of Darwinian selection. Alternatively, in the case of acquired resistance, the cells will not harbor the resistance at the start, but they actually change and acquire the resistance on treatment. And this can happen either in a way where, or in a situation where certain cell clones do harbor other features which allow them to adapt more rapidly and more easily acquire the resistance, or to longer persist in the presence of the drug and survive. So therefore, these cells are also often called drug-tolerant persister cells. However, it is also possible that this acquisition of resistance might happen entirely stochastically, meaning that every cell clone in the population at the start of the treatment has the same chances or probability to acquire the resistance. And which of these situations is true is actually very important to be aware of for the development of additional therapeutics, which will have the goal to prevent or delay the outgrowth of resistance. So, for example, in the case of pre-existing resistance, the aim would be to erase these fully pre-existing resistant cells before they even get selected. In the case of acquired resistance, the goal would be to block the acquisition process. So either this could be in the case of the stochastically acquired resistance, ways that will try ways or strategies that will aim to prevent cells from adapting and changing. So you can think of epigenetic regulators where you could interfere with, while in a situation of specifically acquired resistance, these persister clones might have additional vulnerabilities that could be targeted, for example, specific metabolic dependencies. And we were very interested in this question, if resistance is acquired or pre-existing, and we were thinking of ways to address this. And we like to think of the most naive and simple way to address it. And this would be to actually directly compare a founding clone of a later on resistant clone to its resistant counterpart. As a direct comparison of the same clone before and after treatment could allow us to address if the cell clone changed or if it was the same already at the start of the treatment, indicating it would be pre-existing resistance. The problem hereby, however, is that we only know which founding clones to look at after the resistance has been generated. So what we would need essentially is a way to go back in time and look at the resistant founding clone in the second step after generating the resistance. So we, so to say, would need to have some sort of way to look back into the past or have a time machine. And we actually thought of developing a method which allows to not only identify but also isolate such founding clones and to specifically isolate them alive, because this will allow us to use these clones in functional assays later on also. And therefore, we can then perform a direct comparison of the same clone before and after the treatment, asking, is the resistance pre-existing or acquired? And to fulfill this, technically, we would need to fulfill two prerequisites. The first one would be we somehow need to be able to keep track of the clonal identity within a large population over such a selection experiment. And this we can actually easily do using established lineage tracing methods like genetic barcoding. So in genetic barcoding, short DNA sequences are being introduced into the genome of our cells in a way that every cell clone will receive a different barcode. And therefore, we can then read out these barcodes using next-generation sequencing, and we're always aware of the identity of a cell and all of the progeny of the cell. The more tricky part, however, is how can we identify a specific cell clone within the large population and specifically isolate it by its barcode. And our solution to this was to couple the barcode to a inducible reporter system, as this system or this setup will allow us to not only use a barcode as a unique identifier, but also as a clone-specific switch. So therefore, we can basically turn on the expression of a reporter gene, in this case GFP, but in a barcode-specific manner, which will allow us to detect only these clones within barcode of interest by fluorescent activated cell sorting, and then we can actually sort these cell clones alive. So how this would work is that in the basal state, our reporter, where the barcode is coupled to this GFP, will be off, so there will be no GFP expression. If we then want to activate the reporter, we can make use of the CRISPR A factor dead Cas9 VPR. So this is a catalytically dead version of Cas9, which is fused to VPR, which is a strong transcriptional activator. And in the absence of a guide RNA, this protein complex will not have an effect on our reporter and will not influence GFP expression. However, if we transduce a guide RNA that is compatible to our barcode, this guide RNA will recruit that Cas9 VPR to the locus and specifically induce GFP expression. And most importantly, this happens in a sequence-specific manner, meaning that if the guide RNA is not compatible to our barcode, so it cannot bind this barcode, it will not recruit that Cas9 VPR to the GFP locus, and the reporter will remain off. And this approach we termed CaTCH, which is short for CRISPRa Tracing of Clones in Heterogeneous Cell Populations. So now I would like to guide you through how a CaTCH experiment would work. So we would start out with a cell line of interest, which we would then stably transduce with a dead Cas9 VPR in the first step. And in the second step, we would transduce the cell line with the barcode reporter library where every barcode is coupled to this inducible GFP promoter. And these cells also harbor, or this construct also harbors a constitutively expressed BFP to select for the presence of the reporter. And these barcoded cells can then be used in a selection experiment to select for a phenotype of interest. In our case, this would be therapy resistance. And the surviving cell population can then be, or the barcodes of the surviving cell population can then be read out by next generation sequencing, so we can identify the barcodes of the clones that are capable of growing in the presence of a drug, meaning they are fully resistant. To then retrieve the founding clones of these resistant clones from the starting population, we can go back to a copy of the population which we had initially maintained by freezing, for instance. And this population has never seen any drug, so therefore it's truly treatment naive. And to then aim for the isolation of the founding clone, we can now use the information we obtained in step 3 before and aim for the isolation of the clone with the barcode number 4 in this example. So we transduce the entire population with a guide RNA against barcode 4, which will exclusively lead to reporter activation and GFP expression in this clone. And then we can sort this clone from the entire population and make a cell line out of it. Functional studies. Excuse me. So when developing this tool, we wanted to first test a few things, where the first one was we wanted to be aware of the best possible placing of the barcode in relation to the minimal CMV, which is the inducible promoter controlling the GFP expression, with the goal that the perfect placement of the barcode should lead to the best possible reporter induction and activation being measured by GFP expression. Therefore, we designed a construct that harbors several different specific barcode sequences, which can all be targeted by guide RNAs. and they all are placed at different positions in relation to the promoter. And we then consecutively targeted these different barcode sequences with guide RNAs to measure which position of the barcode leads to the best reporter activation. So here you can see the result of this experiment where we measured the GFP signal after activation of the barcode using different guides. And on the left side, you see the distance indicated of the position where the guide RNA will bind. And the distance is in relation to the transcriptional start site, which is placed within the minimal CMV promoter. And what you can see essentially is that the closer the barcode is placed to the minimal CMV, the better the GFP expression and the activation of the reporter will be. To exclude certain effects or certain effects introduced by the sequence of the barcode, so sequence-specific effects, we also performed the same experiment with a different construct where we shuffled the position of these different barcode sequences. And here we could then also see that essentially the result was the same, that the closer the barcode was placed to the minimal CMV, the better their activation, which we could also quantify. And we identified the optimal window in this reporter assay as 60 to 80 base pairs upstream of the transcriptional start site, as this will lead to the most cells being activated and the highest GFP signal. We also wanted to test the effect of having several binding sites of a single guide in parallel on reporter activation. And here we furthermore tested also if these different binding sites are beneficial, or if it's beneficial to have these binding sites spaced, or if they can actually be directly placed next to each other. And in this assay, we found first that in the spaced construct, so R1 in this example, the signal was always superior over the R2 construct, meaning that having a space between your two binding sites for the guide RNA or three binding sites for the guide RNA is beneficial for the activation. What we also found, not really surprisingly, is that the more binding sites you have, the better the activation will be. So meaning 3 binding sites with SG2 in this example will be better than having 2 binding sites, and 2 binding sites which are relatively or which are close to the minimal CMV promoter are better than having 2 binding sites which are more distal to the promoter. So taking now these results from these experiments, we came up with a final design for our barcodes, where we designed the barcodes as an entire barcode cassette, where the barcode cassette consists of 3 independent short barcode sequences. And these 3 barcode sequences can all be used as binding sites for different guide RNAs. And here we also designed these barcodes in a semi-random fashion, meaning that we do not specifically design the sequence and it's generated randomly. But this still allowed us to include certain base restrictions to make sure that we have the pump site present, which is necessary for the Cas9 to bind. And also we included specific base restrictions at different positions to assure that even though these barcodes are generated largely randomly, that they are still good binding sites for guide RNAs. And the advantage of having a barcode cassette design is that when activating the reporter from such a barcode, we actually have the option to now use and target different barcode sequences in parallel. So for example, in this example, we would target barcode 2 and barcode 1. And the parallel targeting of these 2 barcodes will lead to a stronger reporter activation, which you can see in the top right. And the benefit of this is that a stronger signal will make it easier to distinguish truly activated cell clones from the unactivated population, making it easier to sort these cells. So with this design, we then cloned a barcoding library, and this plasmid library we then sequenced by next-generation sequencing to determine the complexity of the library. And we could identify 130 million unique barcodes present in the library, and these barcodes are evenly distributed in the library. And this complexity is easily enough to label around label a few million cell clones uniquely. So having now generated this tool, we first wanted to assess in a controlled setting if this tool is now actually feasible to trace the evolution of resistance to targeted therapy. And therefore, we designed a proof of principle experiment in which we mimicked a situation where already fully resistant or rough-eye resistant cells pre-exist in a population of cancer cells in a small ratio. And to do this, we engineered a cell line to harbor a single known barcode, in this case barcode 4. And these cells are also, in addition to the barcode 4, labeled with a RFP signal, and they were engineered to overexpress a mutant version of NRAS, which is known to be resistance-causing to RAF inhibition. And these cells with the known barcode 4, the NRAS mutation, and the RFP label were then spiked into a background population of complexly barcoded cells, which are RAF inhibitor sensitive and are, of course, RFP negative and do not harbor the barcode 4. And when we then treat this cell population with RAF inhibitors, we can just use the RFP label as a control and a readout by fax and see that after 30 days of treatment, the entire population essentially consists of RFP positive cells, indicating that our spiked in cell clones indeed take over the population in the presence of the drug. And we can also sequence the barcodes then at the endpoint of this experiment, and we see that indeed the RAF inhibitor treated population consists exclusively of barcode 4, while the vehicle treated population, the barcode 4 still stays at a lower abundance. Now, the more difficult part would now be, or the more interesting part would be, to see if we can actually retrieve these spiked cell clones then with the CaTCH reporter from the starting population. And to do so, we took the starting population in the untreated state and transduced it with a guide RNA against barcode 4. And indeed, we see that only if the spike in population is present and the guide RNA is specifically targeting the spike in population, you see on the right plot that we can detect a small GFP positive population of cells. And using our RFP label, we can actually control in this experimental setup if these cells are the correct ones we are targeting here. And indeed, we can see that all of the cells that turn on the GFP expression are also RFP positive, indicating that we are really targeting the correct cell clones here. We can then sort out the GFP positive cells and expand them in vitro and then sequence their barcodes after this isolation by next generation sequencing. And indeed, here we also see that the barcode of the isolated cell line is exclusively consisting of barcode 4, so the correct targeted barcode, indicating that our isolation procedure was successful. So knowing that the tool in principle works, we next wanted to assess how rare a cell clone can actually be in a population and while we're still able to pick it up with our CaTCH method. Therefore, we performed, again, a very similar experiment where we engineered, again, a cell line with a single known barcode. I, again, used barcode 4 to indicate this. And the cell line also expresses the RFP signal in addition. And these cells are then spiked into a complexly barcoded background population, which is RFP negative and does not have the barcode 4. And we can do the spiking then in different ratios. So essentially titrate the number of cells that we spike in and measure if we can still detect GFP positive cells after transducing this population with a guide RNA against barcode 4. And we can also then assess if we are activating the barcode of the correct cells by the RFP label. So all targeted cells should be GFP positive and RFP positive. So I'm indicating, or I'm showing you here now the results of this experiment, where I'm highlighting the spiked in cells in red, which I can do since I have the RFP label, and the background population is shown in gray always. And when I transduce those two individual populations with the guide RNA4, we see that the background population cannot activate GFP expression, while the spiking population is capable of doing so. And when we then mix these barcode 4 cells in a ratio of 1% into our background population, and this mixed population we then transduce with the guide RNA4, we see that we can now detect a population of GFP positive cells, and these are also all RFP positive. And we can now do this with different spiking ratios. And what we found is that even at a spiking ratio of 0.001%, we still detect GFP positive cells, which are also all RFP positive. So we can basically conclude that we can correctly activate the targeted cells, even at a very low ratio, and the activation efficiency, so meaning the percentage of GFP, of RFP positive cells that turn on the GFP, remains stable across the different spiking ratios, meaning that we have the same activation efficiency even at 0.001%. So while we were developing this tool, also other solutions for the same biological question or the biological problem of the origins of targeted therapy resistance were proposed. And therefore, we wanted, and one of those proposed methods also was based on a technology using barcode-specific reporter activation. And therefore, we wanted to compare this different approach to our approach to see if we can hold up with the alternative approach. And just as a short reminder, the CaTCH approach, which is shown in red here, works by first engineering a cell line to stably express that Cas9 VPR and a barcode-coupled reporter. And in a third step, we then transduce a virus with the guide RNA to induce activation of the GFP expression from the reporter. While the alternative approach works in a way that they express a single guide RNA and use this guide RNA as the barcode essentially, and then in a second step to induce reporter activation, the cells are then transiently co-transfected with plasmids harboring once the dead Cas9 VPR and once a reporter plasmid where the compatible sequence to the guide RNA is placed next to inducible promoter. And to compare these two methods, we essentially performed the same experiment again, so engineering a specific cell line with a single known barcode and an RFP label, and these cells were then spiked into a background population. And what we found in this experiment is that both methods work reliably well, meaning that we can activate the reporter of the correct cells with both tools. However, what we found is that with the transient transfection-based approach, so the alternative approach, we fail to detect cells at a very low ratio, while CaTCH was still able to do this, which we think is most likely due to the transient transfection-based approach of the alternative method. And therefore, while both tools work equally well, the alternative approach fails to detect very rare cell clones, while we are still capable to do this with CaTCH. And here I would also like to point out that recently several other clonal isolation methods using genetic barcodes and barcode-specific reporter activation have been proposed and are published either in peer-reviewed journals or in preprint articles. And they all work with a similar principle, where some of them, equally to our tool, make use of CRISPRa and that Cas9 VPR induce reporter activation, while other tools make use actually of a catalytically active Cas9, which cuts a reporter gene to repair a frame shift, again in a barcode-specific manner, and this frame shift repair will lead to the expression of a functional reporter gene. Last, also in the bottom right, I show you a tool that was recently, or I sketched out a tool that was recently developed by the lab of Arjun Raj, which actually makes use of expressed barcodes, which are then targeted with RNA fish probes to highlight the barcodes in fixed tissues. yeah, and it's going to be interesting to see how all of these tools will compare in the next few years. And I'm highly confident that they will be very compatible in the future to each other. Okay, so back to CaTCH. After having generated this method and first characterized it to see if it is actually working well, we now wanted to apply it in an in vivo-based setting to actually study the evolution of resistance to targeted therapy. Excuse me. And for this experiment, we took a mouse melanoma cell line, which we barcoded with the CaTCH components, and then used an injection-based mouse melanoma model, where we take these barcoded cells and subcutaneously inject them into black 6 mice, and then treat these mice with a combined RAF inhibitor, MEK inhibitor therapy. So just as a reminder, this hits the MAP kinase pathway at 2 nodes. And after, and we treat these mice then until resistant tumors grow out, and these resistant tumors we then excise and generate cell lines out of those and sequence the barcodes. So here I first show you the tumor growth curves during this experiment, which you can see at day 0, this treatment was started, and all of the treated tumors in red regress on therapy, and indicating that the therapy is working and all of the cells are responding. But after 20 to 30 days, fully resistant tumors grow out on therapy. And these resistant tumors were then excised, and we sequenced their barcodes. And first I want to show you here the number of unique barcodes which you could identify per tumor or sample. Excuse me. Where in the black bar in the reference cell line, so this is the cell line that we initially injected, we had roughly 60,000 unique barcodes present. And in untreated tumors, roughly half of those, so around 30,000 barcodes, were still maintained. And very strikingly is that the resistant tumors were very strongly reduced in the clonal complexity, with only 1,000 to a couple of thousand barcodes being detected per tumor. And what is even more striking is that if we look at the clonal composition of these tumors, we see that the resistant tumors are almost clonal, and they're mainly driven by 2 to 6 main clones. So how I'm indicating this here is that each colored section of a bar here represents the proportion that a single clone makes up of the entire tumor. So for example, at the far right, you see the dark green proportion, which would be a single clone or a single barcode, which makes up roughly 80% of the reads of this tumor. And all of the white areas are very lowly abundant barcodes summed up, indicating that in the reference cell and in the untreated tumors, there are no barcodes specifically enriching. So having now found several clones which highly enrich in resistant tumors, we could now select some of those clones for clonal isolation using CaTCH. And we aimed to isolate these clones once from the treatment-naive population, so the starting population, and once also from the resistant population to really generate a clonal cell line. So what I'm showing you here now is the distribution of barcodes in the treatment-naive starting population, where these barcodes are ordered by their abundance, with the most abundant barcode being on the left side and the least abundant barcode being on the right side. And in the colored dots indicate now the initial abundance of the clones which we picked for isolation using CaTCH from the treatment-naive starting population. And after sorting these clones, we then sequenced again the barcodes of now the purified cell lines, and we can see that we actually can strongly enrich for those barcodes, where some of these clones, clones or barcodes, could then be detected at a purity of 90% or higher, with a fold enrichment being indicated in blue here. And we can then also depict more or less the same results in the bar graph view, which I explained to you before, which I now use to highlight that we basically could generate several clonal cell lines at a high purity, now from the treatment naive and from the resistant tumors, with matching barcodes. So leaving us with now several clonal pairs in the treatment-naive and in their resistant state. And this is not a perfect model to actually compare these clones and first ask if the resistance is actually pre-existing in the treatment-naive clone or not. And to test this, we essentially performed a very similar experiment as the previous in vivo experiment. But now we inject the clonal cell line of either only the treatment-naive or only the resistant clone, again subcutaneously into Black 6 mice. And then we measure the drug response by treating these mice and assessing the tumor growth by caliper measurements. So here I show you the results of this experiment for a single clonal pair. In this case, it would be barcode 121, where in red, I always depict the growth curve of the resistant version of the clone, where you can see here that this clone really does not respond to the therapy at all, indicating it's really stably and truly resistant. And in blue, I show you the growth curve of the treatment-naive version of the clone. In gray, I highlight the untreated growth controls of these clones in this experiment. And what we see here is now that the treatment-naive clone actually does respond to the treatment and regresses, indicating that it is not resistant from the start and that the resistance was acquired later on. And we performed the same experiment now with several other clonal pairs and essentially found the same result, meaning that all resistant tumors grew unperturbed in the presence of the drug, while all of our treatment-naive clones in blue here actually respond to the drug, indicating that the resistance is not pre-existing. The fact that most of these clones do grow out on drug later on and actually do form resistant tumors eventually is something that is not, or seems to be something that is not specific to these clones that we isolated here, as we also isolated control clones, which were specifically chosen by their absence of resistant tumors in the previous experiment. So they were specifically selected to be sensitive to the drug, and even those clones acquire resistance at one point after prolonged treatment. And we can also perform the same experiment with several randomly isolated single-cell clones, and also all of those will acquire resistance at one point, which tells us that resistance seems to be a feature that is universally achievable in the cell line that we're using here. However, we found one clone which in its treatment-naive state actually showed a reduced response to the drug and resumed growth on therapy just 3 to 5 days after treatment. And this is very interesting to us because it could indicate that there is indeed some feature of pre-existing resistance or reduced response to the drug. Therefore, we wanted to look a little bit more closer into this clone and what's going on there transcriptomically. And therefore, we explored QuantSeq 3' mRNA sequencing, which is our standard method of choice for mRNA sequencing. And we sequenced this clone now after injection into a mouse and after tumor engraftment. And then harvested some of these tumors and sequenced them using QuantSeq. And we also sequenced some tumors which were treated for 3 days with a combined RAF inhibitor-MEK inhibitor therapy, which we then also excised again and sorted out the cancer cells by their GFP label and also performed mRNA-Seq. And the first thing we saw here is that if we now perform a upstream regulator analysis on these clones, we see that our treatment-naive version of our clone with the reduced response, so this is 2646 in this example, already has similar programs activated as fully resistant clones to after prolonged treatment, while a other treatment-naive clone, in this example 14559, who showed a longer response to the drug, did not have these programs activated. And we can also more closely look at the expression levels of specific genes being having been previously reported in causing resistance to targeted therapies. And here we see that several of these genes are readily up-regulated also in the 2646 clone in its treatment-naive state, similar to later on fully resistant clones will upregulate these genes. Also at the bottom, we see here the expression levels of the melanocyte master regulator MITF, which shows low levels in our T2646 clone, which has previously been linked to a increased clonal plasticity in these cells, and has also been linked to reduced response and increased resistance potential to RAF inhibition, potentially explaining what is happening in this clone here. Last, we also wanted to look at genetic alterations which might be responsible for the resistance in some of our clones. So therefore, we performed whole exome sequencing on a set of several clonal pairs, and we found in one clone in the resistant form a mutation in KRAS at the residue G12. And this is very interesting because G12 mutations in KRAS have been previously described to be activating for KRAS and have been linked to being causative for resistance to MAP kinase pathway inhibition, or more specifically RAF inhibition. And the interesting thing here is that the treatment-naive form of this clone did not harbor this mutation. And as you can see again on the left, you see that this was a clone which really responded for a long time to the drug in the treatment-naive state. So therefore, this provides as experimental proof of the de novo acquisition of a potential resistance-causing mutation. So with this, I would already like to sum up and hope that I could convince you that we have a new cool lineage tracing tool at hand, which can be used to study the clonal evolution of resistance to targeted therapies. And we also envision that future applications of CaTCH can actually be used for other evolutionary processes and clonal selection processes, for example, the evolution of metastasis or reprogramming events. And with this, I would like to come to acknowledgement slide. And of course, first of all, thank my PI, Anna Obenauf, for the cool project and the ongoing support, and the entire lab, and former students for helping out on the project, lab sharing, lab space sharing labs, the great facilities we have at the IMP, also my PhD committee, which always provided great advice and help on this project, and the coordinators of the program. And yeah, thank you very much for your attention. And I would now like to hand over to Martina, who's going to tell us a little bit about QuantSeq. Thank you.
So thank you, Christian, for this really, really interesting talk. I'm super impressed. I'm very happy that we could contribute to your work here. So to give Christian a short break before you bombard him with questions in the Q&A session, I just want to wrap up a little bit about QuantSeq, which Christian has used in the study to characterize the transcriptome of his identified cancer treatment-resistant cell clones. And QuantSeq was particularly suitable for this kind of analysis, to have a fast access to the transcriptome, since it generates only one fragment per transcript, in contrast to common mRNA-Seq methods, which generates many fragments per transcript, and you have to assemble in silico afterwards to get the data. QuantSeq only generates one fragment per transcript, and this enables you to have fewer reads per sample required for having a meaningful expression profiling, and it also enables higher level of multiplexing. And the data analysis downstream is super straightforward, so that the wet lab scientists themselves can analyze the data without big need in bioinformatics. And a second advantage of QuantSeq over common mRNA-Seq methods is that it's super fast and very easy to use for gene expression profiling. It consists of only 5 major steps in a very streamlined protocol. You don't need any pre-processing steps like mRNA enrichment or ribosomal RNA depletion. You go straight for polyadenylated mRNAs, and this obviously saves time and consumables. And if you're going for larger projects where you want to screen for whatever clones characteristics or drug screenings, we now have QuantSeq-Pool since almost a year on the market. QuantSeq-Pool is the tool of choice for large screening projects because you can pool up to 96 reactions after the very first step in the library preparation protocol and pool them into one reaction and process up to 96 samples in one tube downstream for the library generation. And this, of course, saves a lot of time in QC and in preparation of the RNA-Seq one, but also it reduces the technical variability. And obviously, you save consumables, so you just need one reaction instead of 96. So ultimately, if you combine this with UDIs, you can multiplex over 36,000 samples per lane if the sequencing capacities are growing even further. So that was just a short reminder on what we offer for QuantSeq-Pool and why we supported this webinar. And if you have any questions about QuantSeq or QuantSeq-Pool, you can contact our support team to consult about your project. If you're interested in an introductory offer, please send us a message at sales@lexogen.com or visit our new website, QuantSeq Intro. And with that, I'm handing back to Julia and have fun in the Q&A session.
Yeah, thank you very much, Martina. Also, thank you to Christian for that very nice presentation. As a reminder, if you have a question, please type it into the Q&A panel. And we would also like to ask our attendees to take a moment after the webinar has ended to take our exit survey and provide us with your feedback. So with that, let's see what we have here for questions. Okay, here's one for Christian. How long does it take between injecting the barcodes and turning on the reporter in vivo? And is this cell type dependent or is it affected by the proliferation rate?
So how we do the experiment is essentially we barcode our cells in vitro. You have then several purification steps to basically select for only barcoded cells, and these you then need to raise up. So this basically depends on, or the timeline of this depends on how fast your cells are actually proliferating. If this correctly answers your question. Because the reporter activation in the end is then actually only performed again in vitro with the cell line that was generated in vivo. And here we basically transduced in the entire cell line with the guide RNA against the barcode, and this takes roughly a week until we measure the reporter activation in these cells and then sort these out. And since we only sort out a very small number of cells, then again the timeline until these cells are grown up again takes a little bit and depends on the proliferation rate of the cells. Again, for cell type dependent, I'm not sure if, or I'll try to answer it. Please just write again if you're not sure if I'm answering it the right way. But here we have seen that so far the system worked in every cell type that we tested. So the reporter activation there does not seem to be in any way dependent on the cell type or the proliferation rate per se. But of course, I can imagine that very tricky cell lines, which grow very slowly, that are just in general difficult to work with, will also be difficult to work with this tool, as you do have those sorting steps and prolonged times where the cells do need to grow.
Okay, great. Yeah, that's all good to know. Somebody else wants to know if you have performed any single-cell DNA sequencing, for example, to detect rare clones.
No, we have not done this. This I can answer very shortly.
Okay, that's an easy answer. Is it possible to do single-cell RNA sequencing with CaTCH?
At the moment, it is not yet possible, or at least, I mean, you can, of course, do single-cell RNA sequencing, but you will not obtain the barcode information because the barcode is only present as DNA in our cells. But we're currently working on a follow-up tool, which also will make use of expressed barcodes, which can then be detected in single-cell RNA sequencing, making our CaTCH tool compatible with single-cell RNA sequencing as well.
Okay, great. Thank you. Let me see. So the CRAS mutation you detected in that one experiment you mentioned near the end, how common is that? How often is it identified in a resistant clone?
So in the presentation, I only showed you now the result for one clone, but by now we have characterized a couple of more of these clonal pairs, and we indeed find KRAS mutations more commonly now in our cell model in resistant cells, not exclusively the G12 mutation that we found, but also mutations at other known residues leading to KRAS activation. So this really seems to be something that in our model is reoccurring after prolonged treatment and resistance generation. Why, however, it's specifically KRAS in our cells, which is actually rather uncommon to be mutated in melanoma, while melanoma cells usually harbor alterated mutations of NRAS, is something we do not know yet. Could be an artifact of the cell line model per se, but we are looking into this also currently.
Okay, great. Thank you. Oh yeah, so is the CaTCH method published anywhere? Are you planning to publish it so participants can get more details about the method?
So the method is published. We have a paper out in Nature Biotechnology, Christian Umkehrer et al., which was published last year. So you can find it on PubMed.
Okay, very good. And then another question for you, is the CRISPRa of the reporter affected by epigenetic changes in the genome, for example, methylation of the promoter?
That's a very good question and something we have not looked into yet. It might be something that can happen. Specifically, I could imagine, but again, this is something now just highly speculative. I don't know that specifically in those in vivo experiments with the prolonged treatment in vivo and potentially also the resistance generation, that there could be silencing happening with this reporter. This is something I cannot rule out, but also something we have not characterized. What we have found is that initially in the cell line that is labeled with our CaTCH tool, we do find a certain percentage of cells that is directly after labeling incapable of activating the promoter. But we assume that this is rather the case due to integration-specific effects, as this was described with the doxycycline system, which is per se, or we lend some components of the doxycycline inducible system there also. And this has been shown that in the doxycycline inducible system, this is due to integration-based effects. We, however, circumvent this in the cell line generation by subsorting already a cell line that has a low leaky expression of the GFP, so we can make sure that these cells are in the open chromatin area, and then the actual cells that end up in our assay are not affected by these integration-based effects.
I see. Okay, great. Can you say a quick word about why you chose QuantSeq to characterize your clones?
Sure. I think Martina already summarized it quite well.
Right.
But the reason why we use QuantSeq in this project, but also in several other projects in the lab, is basically that it's very straightforward to use. The protocol is very fast. And if you're interested in mRNA expression, makes it a lot cheaper to use QuantSeq than, for example, entire RNA-Seq, as you need less reads on the sequencer later on. And this will also greatly reduce sequencing costs. So therefore, we like it very much.
All right, that's good to hear from you also. Somebody would like to, is asking whether you could repeat what size of barcode library you used for your in vitro or in vivo experiments. That was probably already mentioned, but that person didn't get that part.
Mm-hmm. No problem. So the barcode library we use per se is the very complex library with 130 million unique barcodes. In the different experiments that we used, we, however, limit the complexity to make it fit whatever the experiment requires. So, for example, in the in vivo-based assay, we cannot work with such a high number of barcodes because not so many clones will even engraft in your tumors. So therefore, we reduced the number of barcodes. And in this experiment, we worked with a complexity of 60,000 unique barcodes at the injection time point.
Okay, great. Thank you. And then finally, you hinted at this at the end, but can you just talk a little bit about what other biological questions you might be able to address with CaTCH?
Mm-hmm. Of course. So yeah, overall, maybe to stay on the field of resistance, you can essentially use any drug treatment that where clonal evolution might be an influencing factor with CaTCH. But again, we think it's not limited to this. So we are also very excited about actually using it in evolution of metastasis, because this is also a topic where the question how metastases are actually evolved is also still largely debated. And therefore, we have already performed first assays with injection-based metastasis models, where we then want to trace back the clones that are capable of forming metastasis at specific sites, and then assess if these clones are somehow prone to do this, or if it is a stochastic selection.
Okay, great. Thank you very much. It looks like this is all the questions we have for today. So let me thank our speakers, Christian Umkehrer and Martina Sauert, and our sponsor, Lexogen. And as a reminder, please look out for the survey after you log out to provide us with your feedback. And finally, if you missed any part of this webinar or you would like to listen to it again, we will send you a link to an archived version via email. With that, thank you very much for attending this Genome webinar.
Thank you.